Our next speaker is Gerardo Lederkremer from Tel Aviv University, and he will talk to us about Huntington's. Please.
Okay, thank you, Miguel, for inviting me, and I'll tell you about our studies on Huntington's disease. So, Huntington's disease is on the limit of the rare diseases as a genetic neurodegenerative disease characterized by these typical movements called Huntington's chorea. Also, there's cognitive impairment. We became interested in Huntington's because it shares a lot of common features with other neurodegenerative diseases, but it's monogenic, so the cause is very clear, but the pathway to cytotoxicity was much less clear. So in Huntington's, you have this mutation, which is an expansion of a CAG repeat that codes for glutamine. So you have this polyglutamine stretch in the huntingtin protein. If the stretch is less than 35 glutamines in length, the protein is normal. And if it's longer, it causes the disease. And the age of onset of the disease is inversely proportional to the extent of the repeats. The longer the repeats, the younger the age of onset. As other neurodegenerative diseases, it develops first in a specific region of the brain, in this case the striatum. Not clear why. There's no cure or effective therapy so far. As I said, there's a motor and cognitive dysfunction, progressive with age, and death in average about 10 years after onset. So to try to understand better the cytotoxicity in the disease and see if one can develop a therapy, as you know, in drug discovery we have two approaches. One is the unbiased, naive, and the other one is hypothesis-driven, target-based. And in recent years, one can see here in blue, there has been much more success in the hypothesis-driven. These are the number of approved first-in-class drugs in recent years. Okay, so much higher success. So we try to understand, again, better the pathway of cytotoxicity. So what does Huntington's have in common with other neurodegenerative diseases? One is the aggregation of the huntingtin protein, and this is similar to what happens in Alzheimer's, Parkinson's. We heard about ALS, prion diseases, etc. So formation of insoluble aggregates, also called plaques, inclusions, etc. So the aggregation. Another thing that has been found in recent years to be common to all these diseases is the development of endoplasmic reticulum stress, ER stress. And we know from cell biology studies that a prolonged ER stress leads to cell death. And the question, was one of the questions was how important is ER stress in the development of the cytotoxicity in Huntington's as a model. And if we can inhibit this ER stress, or the path between ER stress and cell death, as a form of therapy. Obviously, the first approach that was tried was to try to inhibit the aggregation itself. But this hasn't succeeded so far. Several companies have also tried to inhibit the aggregation of Huntingtin, and this hasn't worked, not in Huntington's disease and not in any of the other neurodegenerative diseases so far. And perhaps explanation comes from the fact that as we and the others have found, the actual formation of these insoluble aggregates is a last resource form of protection of the cells. It's not the aggregates, the final aggregates are not toxic. What is toxic is the pathway towards the aggregates, the association of the misfolded proteins into toxic oligomers. In our case, we could link the development of ER stress directly to the formation of the oligomers, not the aggregates. So if one tries to inhibit the aggregation, it's very probable that one will stop this last resource protection, packing of the misfolded proteins into these large aggregates, and that could cause more damage than help. Okay, so what's ER stress? ER stress is defined as accumulation of unfolded or misfolded secretory proteins. Simply that. This appears in a series of conditions in where one affects the folding of the proteins. One is that if we have expression of mutant secretory proteins that they misfold, but also upon synthesis or requirement of synthesis of large amounts of secretory proteins, there can be ER stress, conditions of oxidative stress, hypoxia, calcium imbalance, glucose starvation, and inhibition of ERAD, of the ER-associated degradation of the secretory proteins. Secretory proteins are synthesized, and a large percent of the molecules are sent constantly to what's called ERAD. For some proteins, I'm talking about wild-type proteins, very low percents make it out of the ER and to their final destination, like the plasma membrane. So if one inhibits ERAD, the proteins accumulate, and we get ER stress. In a simplified manner, we have sensors that are membrane proteins on the ER membrane that sense the presence of unfolded proteins in the ER. So in the top here, we have the ER lumen. Here, there's one of the sensors, the IRE1, associated with this chaperone BIP that keeps it in an inactive state. But if there are many unfolded proteins in the ER, they associate with BIP, which dissociates from IRE1. IRE1 dimerizes then and becomes activated. And then it activates a series of downstream responses. Now, where does huntingtin come in here? Huntingtin is a cytosolic protein, as is SOD1 or as many other proteins that misfold in the cytosol, and they cause ER stress. So how does this work? So what happens is that during the aggregation process, there's an inhibition of proteasomes. Inhibition of proteasomes inhibits these pathways to ERAD. That means the degradation of proteins that come from the ER and go to the cytosol for degradation. The accumulation of those proteins then causes ER stress. and prolonged ER stress by the accumulation of these unfolded or misfolded secretory proteins in the ER, or actually in this compartment that we have described, the ER quality control compartment that I won't have time to talk about. This persistent or prolonged ER stress leads to cell death, apoptosis. And this would cause neurodegeneration. So just one, I would say, quite dramatic result that illustrates the timing of when the aggregates, the insoluble aggregates of the huntingtin form and when the ER stress develops. So here on the left side, we would have an ER stress marker, which is simply a model misfolded secretory protein and the ER, and it shows at the beginning this ER pattern. And here in the middle, if you see any signal, there's a dispersed huntingtin protein, and this is, so here we're looking at the model huntingtin with 96 glutamines, pathogenic, linked to cherry fluorescent protein, and the ER protein linked to GFP. And if we look with time, so let's start this. I don't know if you can see that there's not much change here, but you can see already a redistribution here and an increase in the signal in this region. So the ER protein, the ER stress marker is accumulating. The huntingtin doesn't change much, and suddenly we get the formation of the huntingtin aggregate that absorbs all the huntingtin from the cell, and we have the formation of this large aggregate. But clearly, the ER stress develops before the formation of the large huntingtin aggregates. So what would be happening is in this model where we have the ER and proteins being sent to ERAD, to degradation at the proteasomes from the ER. Here we would have the huntingtin monomers well folded for the wild type huntingtin. If we have the presence of a misfolded huntingtin, it would form these toxic oligomers. They inhibit the proteasomes. The ER protein accumulates, causes ER stress and induction of what's called the unfolded protein response, UPR. And this causes a series of downstream events, which are the UPR. And in the long term, this leads to mitochondrial apoptosis, through the mitochondrial pathway. At some point, the oligomers, they associate into polymers and form these large aggregates, which are the amyloid fibrils that we heard about, which are a common structure for all these diseases, like the A-beta and Alzheimer's, or like the synuclein and Parkinson's, etc. And we heard from Udi Gazit that there are many new diseases that also involve the formation of these amyloids. They all have the same structure. Even if they're very different proteins, they all have the same structure. And in these structures, the problematic regions here in blue are protected. And so these aggregates, these large aggregates, the amyloid fibrils, are less damaging to the cell. And in this case, they would not be causing ER stress anymore. So we talked about prolonged ER stress. What happens at the early stages? So at the early stages, the unfolded protein response tries to solve the situation. And one of the ways is by transient inhibition of protein synthesis. If the protein synthesis in general in the cell is inhibited, there's no more feeding of misfolded proteins that create ER stress. The other is upregulation of chaperones that try to fold better the secretory proteins. Upregulation of the ERAD and autophagy machineries to try to degrade more of these secretory proteins. So all these pathways leading to protein homeostasis. And if this succeeds, all is okay. But if they don't succeed in the long term, this initiates apoptosis. Without getting into the details, just to show the complexity of the three different pathways and mammalian cells of the unfolded protein response. And here in numbers, I put the different stages. So there are factors that are upregulated very soon after the accumulation, after the initiation of ER stress, others later. And at the end, you have the upregulation of pro-apoptotic factors, for example, this one, CHOP. What happens, going back to Huntington's? So, as I said, the most sensitive region of the brain is the striatum. And what happens in striatal neurons? We try to look at the striatal neurons compared to other cells. And what we saw is that of the three pathways, the one that was most affected and in a different way was the PERK, what's called the PERK pathway. PERK is a kinase, again, sitting in the membrane of the ER. And what it causes is the phosphorylation of this protein synthesis factor, EIF2 alpha. And that way, it inhibits transiently translation in the cell. In the long term, the same pathway causes upregulation of pro-apoptotic factors as CHOP and apoptosis. So if we look at striatal neurons expressing wild type or pathogenic huntingtin, one can see that the phosphorylation of EIF2 alpha is much higher with the pathogenic protein. We could see something similar when we looked at sections of wild type or Huntington disease model brain. And we could see that in the striatum, the concentration of this IF2 alpha and the phosphorylated form was very low and was much upregulated in the HD model. And so we determined that this actually seems to underlie the sensitivity of the striatal neurons to the huntingtin. And why do we say this? Because one thing we thought that we could try to do is to inhibit this pathway. And by inhibiting this phosphorylation in striatal neurons with the pathogenic huntingtin, we could actually rescue the cells from cell death. We then made a collaboration with Moshe Portnoy, a chemist here at the Tel Aviv University, that made some derivatives of a known PERK inhibitor of this UPR sensor kinase. The known inhibitor had been found just by screening inhibitors in vitro from the Cambridge library, chemical library. And some of the compounds were actually much more active than the original compound, like this one that we call MK28. Here I'll show you one assay. We're looking at apoptosis with the addition of a small amount of this additional ER stress with this compound tunicamycin. So this is apoptosis without the drugs. And here we titrate increasing concentration of the drugs. This is the original drug, and this is the MK28 derivatives. So very good and inhibiting cell death caused by ER stress and these striatal neurons expressing a pathogenic form of the huntingtin. What about without an ER stressor? So this is simply cells grown without serum to stop cell proliferation. And we can see this, the original compound doesn't do too much, but the derivative lowered very significantly cell death. So we tried to see if this works in HD model mice, and we looked at a very severe model, the R62, simply because these mice, they die they show symptoms very soon, and they die in about 100 and something days. And this was in collaboration now with Danny Offen at Bellinson. And what we could see, the way we did it is we applied subcutaneously these dialysis pumps, Alzate pumps, and we delivered the drug continuously then for just 4 weeks. And after those 4 weeks, we see the effects. And I'm showing just one of the tests of motor function. In this case, vocalizations. They have some effect also of cognitive function. And we can see the transgenic mice and the mice that were given the drug for 4 weeks, a very significant improvement. And if we look at the survival of the mice, and this is much after the drug was stopped, right? Still we have a nice effect in extending the onset of death of the mice. So the strategy, again, is to inhibit an ER stress pathway, in this case the PERK pathway, interfering with the cytotoxic effects of the huntingtin. This might work as a generic approach in other neurodegenerative diseases where also ER stress is very determinant. And for example, as a proof of principle, it was shown that if one inhibits the eIF2 alpha phosphorylation in some Alzheimer's models, one gets significant amelioration of the symptoms. So this was done mainly by Julia Leitman and Marianne Schenkman, my lab, Javier Gans in the collaboration with Danny Offen, also Hagit Eiger, Ori Yankovici. And these are collaborators in initial stages of the studies, Ulrich Hartel at the Max Planck, and Uri Asheri here, and more recent stages, Moshe Portnoy and Danny Offen. Thank you.
Thank you, Gerardo. Questions? One question.
So as you know, huntingtin is also an adapter for axonal transport production by first to another. So did you look maybe the drug has any effect on transport, axonal transport?
We didn't look at transport. Yes, there are many studies with huntingtin, and it's involved in many different things. We looked only at ER stress, just mainly because, well, first because it's our specialty, and second because we saw a very strong effect of ER stress caused by the huntingtin, which was cytotoxic. So.